Hello. So let's continue with enzyme part two. So today we'll be looking at enzyme regulation. So enzyme regulation in living systems. Regulation of enzyme activities occurs at different stages in one or more of the following ways to achieve cellular economy. So for enzyme regulation, we have different types of enzyme regulation. So now we are coming to look at how or the different types of enzyme regulation that we have. So I think when you say regulation, in a layman's language, regulation can be simply mean you controlling something. Do you get it? Yeah. So we are coming to look at how the enzymes they control the living system. So the first one is allosteric regulation. The second is activation of latent enzymes, compartmentation of metabolic pathways, control of enzyme synthesis, enzyme degradation, and the last one is isoenzymes. So let's dive into allosteric regulation. So some of the enzymes possesses additional sites known as allosteric sites besides the active sites. So in our previous lesson, we learned that the active site, that's the place or the site where the substrate binds with the enzymes to form a product. You get it? So any other site apart from the active site is known as the allosteric site. Do you get it? Any site apart from the active site is known as what? The allosteric site. So such enzymes are known as allosteric enzymes. Allosteric enzymes. So allosteric enzymes are simply enzymes that have additional sites apart from what? The active sites. So those enzymes are known as what? The allosteric enzymes. The allosteric sites are unique places on the enzyme molecule. So there are different sites altogether apart from the active site for another purpose. So allosteric regulation. Certain substances referred to as allosteric modulators into brackets effectors or modifiers bind at the allosteric site and regulate the enzyme activities. So this is how allosteric regulation occurs. So we have some substances which are known as allosteric modulators or effectors. So those allosteric modulators they will bind at the allosteric site and regulate the enzyme activity. So that's how allosteric regulation occurs. So for allosteric regulation, the enzyme, so we learned previously that allosteric enzymes are enzymes that have additional sites apart from the active site. So in allosteric regulation, we have allosteric modulators, substances called allosteric modulators. So you can search for examples of allosteric modulators. So these allosteric modulators, they will bind at the allosteric site. So this is how the allosteric regulation is occurring. So the allosteric modulators or effectors, they will bind at the allosteric sites, then they will regulate what? The enzyme activity. So the enzyme activity is increased when a positive allosteric effector binds at the allosteric site known as the activator site. On the other hand, a negative allosteric effector binds at the allosteric site called inhibitor site and inhibit the enzyme activity. So this is just simple. So what this is saying is that for the enzyme activity to increase, it is increased when a positive allosteric effector. So we have two types of allosteric effectors. That's the positive and negative. So the positive allosteric site, they help in increasing the enzyme activity by binding to an allosteric site called the activator site, and the other one is the negative allosteric effector, So for the negative allosteric effector, for them they bind at an allosteric site called the inhibitor site and inhibit the enzyme activity. So the next one is feedback regulation or inhibition. The process of inhibiting the first step by the final products in a series of enzyme catalyzed reaction of a metabolic pathway is referred to as feedback regulation or feedback regulation or inhibition. So for feedback regulation or inhibition, is the process of inhibiting. When we say inhibiting, or when we talk about inhibition, it simply means you stopping something to happen. You get it? Like more or less that you are decreasing its activity. So it's the process of inhibiting the first step by the final product. So feedback regulation or inhibition just simply means the process of inhibiting the first step by the final product. So the first step of any process, so in feedback regulation and or inhibition, we are trying to stop the first step of that process by the final products that the process will produce. We are trying to stop the first step of the product by the final um the first step of the process by the what final product. This is a real cellular economy to save the cell from the wasteful expenditure of synthesizing a compound which is already available within the cell. Okay, so what this simply means, with the help of feedback regulation or inhibition, it helps us to save the cell from wasteful expenditure. So instead of the cell to spend on creating other substances or giving out some extra substances, this feedback regulation or inhibition, it simply stops the cell from producing extra compounds which is already available in the cell. So it reduces excessivity of product in a cell. So more or less like for feedback regulation or inhibition in a cell or in the human body, it helps the human body, uh, it stops the human body from producing so many substances which are already in what in a cell. That's all about the feedback regulation or inhibition in our system. So feedback inhibition or end product inhibition is a specialized type of allosteric inhibition necessary for control metabolic pathway for efficient cellular function. Okay, so we have some enzymes with allosteric effectors. So you will get the enzyme on the left part, the metabolic pathway inhibitor, then activator. So what this means is that you see for all the enzymes that they've listed, the metabolic pathway means that the enzyme that will be listed, the metabolic pathway that it occurs or it works in is what it is listed there. So for hexokinase, it works in glycolysis. So the inhibitor for hexokinase is glucose phosphate, and for hexokinase doesn't have any activator. Inhibitor means that glucose phosphate stops the action of hexokinase. Do you get it? But hexokinase doesn't have activator, so it doesn't have anything that will activate its work. Let's go to the second one. That's phosphofructokinase. Phosphofructokinase, it also works in glycolysis. That's the metabolic pathway that phosphofructokinase works in. So phosphofructokinase, it is inhibited by ATP. It is inhibited by ATP because the phosphofructokinase in glycolysis, that will help us to produce more ATP. So if you bring in another ATP, definitely it will inhibit it. Do you get it? But it is activated by AMP or ADP. AMP or ADP activate phosphofructokinase because when the cell is not sufficient with ATP, the cell will be required to produce ATP. Do you get it? So these AMP and ADP will help activate the phosphofructokinase to produce more ATP. But ATP is an inhibitor because when you have excess ATP in a cell, there is no need for the phosphofructokinase to also produce ATP for you. So that's how, or that's what the table is talking about. So we have the enzyme, metabolic pathway, inhibitor, and the activator. Sometimes it normally comes in the exam, so you can take notes of it. Okay, so let's move on to latent enzymes. So for latent enzymes, some enzymes are synthesized as proenzymes or zymogens. So proenzymes are also known as zymogens, which undergo irreversible covalent activation by the breakdown of one or more peptide bonds. For instance, proenzymes, namely chymo, chymotrypsinogen, pepsinogen, and plasminogen, are respectively converted to active enzyme chymotrypsin, pepsin, and plasmin. So what this is saying is that we have some enzymes which are produced as proenzymes or zymogens. So these proenzymes or zymogens, they go under irreversible covalent activation. That means you cannot send it back. Irreversible, it can't go back. Do you get it here? So they undergo some irreversible covalent activation by breaking down peptide bonds. We know that peptide bonds, they are mostly found in proteins. They help in the build up to protein. So for example, some of the proenzymes are the pepsinogen, plasminogen, etc. So these pepsinogen and plasminogen, they are converted to their active sites or their active enzymes. So you can see that pepsinogen is converted back to pepsin. That's its active enzyme. You see, initially they were pro-enzymes. Pro means before. Pro means before. Eu, issue means true. Do you get it? And A means false or no. That's why we have prokaryotes, eukaryotes, and eukaryotes. You get it here. So this one, they are pro-enzymes. They are not yet enzymes. So that means they have to be irreversibly converted into an active enzyme. Do you get it? Yes. So these enzymes, they will be converted to their active enzyme. So their active enzyme, so you can see that pepsinogen could be converted to pepsin, then plasminogen will be converted to plasmin, then chymotrypsinogen, that one will be converted to its active enzyme called the chymotrypsin. So that's what the latent enzymes is also talking about. So it says certain enzymes exist in their active and inactive forms, which are interconvertible depending on the needs of the body. The interconversion is brought about by the reversible covalent modification, namely phosphorylation, dephosphorylation, and oxidation reduction of diphosphide bonds. So you can search for more information on it when you are free. So the next one is glycogen phosphorylase. So for glycogen phosphorylase, this glycogen phosphorylase is just an enzyme which breaks down glycogen to provide energy. Yeah. So for this enzyme, it's being normally found in the muscle. Yeah. So it will just break down every glycogen in the muscle for us to produce energy. So you can read more on that. Normally it doesn't come in exam, but if you need um any extra info about that, you can just search for it. So compartmentalization. There are certain substances in the body, example fatty acids, glycogen, which are synthesized and also degraded. There is no point for simultaneous occurrence of both the pathways. Generally, the synthetic intermediate anabolic and the breakdown catabolic pathways are operative in different cellular organelles to achieve maximum economy.